This is the lecture review for the nervous system. This part covers the introduction to the nervous system, in which we talk about the cell types found in the nervous system, also the overall organization of the nervous system, and the structures that are responsible for the generation and conduction of nerve impulses within the nervous system. In later lecture reviews, we focus on the central nervous system, peripheral nervous system, and some autonomic nervous system structures as well. You can organize the nervous system as either central nervous system, which constitutes the brain and the spinal cord, and the peripheral nervous system, which is everything else, including the specialized sensory structures, as well as your general senses, and all the nerves that are carrying the information to and from. You have a sensory division, also called afferent, that is in charge of sending afferent signals from your peripheral tissues, sensory information from those tissues to the central nervous system. So that's the sensory division. Motor division is the other direction. It's motor signals that are going from motor neurons to give motor signals to either skeletal muscle, any type of muscle, cardiac muscle, smooth muscle, as well as glands. The somatic nervous system is in charge of controlling the somatic motor part of your body, so your voluntary muscles. Pretty much that's it. The autonomic nervous system is everything else. It includes the cardiac muscle, control of cardiac muscle, control of smooth muscle, and any glandular secretions that are produced in response to nervous signals. The autonomic nervous system is composed of two main branches, the sympathetic division and the parasympathetic division, and we'll look at those in a little more detail later. But in a nutshell, your sympathetic division is generally mobilized when you are trying to prepare for a lot of physical activity. So mobilizing energy reserves to enhance that activity. And the parasympathetic is more of a conserve energy status of your body. So your rest and digest mode. You can classify neurons within this context as well. So you can have stimuli that are sensed by receptors, and that signal, that receptor signal, will travel down a sensory neuron. Sometimes a sensory neuron is also the receptor as well, or sometimes there's an intervening structure, such as a specialized sensory structure that is actually receiving the stimulus and transmitting that signal to a sensory neuron. But here we're simplifying things. Here our sensory neuron is also the receptor. And the sensory neuron sends afferent signals, sensory signals, to the central nervous system. Here's a look at the cross-section of a spinal cord. And you see that you'll have that sensory information being now passed to an integration center. And these are basically one or several interneurons. And so you have a sensory neuron and interneuron, and the interneuron in response is sending a motor signal to a motor neuron, and the motor neuron is the neuron that is sending the motor information to the final effector organ, be it a whatever type of muscle or a gland. So this is an important organization, something we to think about the nervous system. And remember, the bulk of the neurons, especially in your brain, are all interneurons for the most part. There's also a lot of motor neurons in your central nervous system. Basically, your sensory neurons tend to be, at least the cell bodies of those sensory neurons are generally located in your peripheral nervous system. So let's look at the cell types that are found in nervous tissue. And of course you have neurons. And we're going to talk about the neurons in a little later here. But the other major type of cell are called glial cells, or neuroglia. And these are all your supporting cells. In the central nervous system, you have astrocytes, microglia, ependymal cells, and oligodendrocytes. In the peripheral nervous system, you only have what are called satellite cells and Schwann cells. So we'll look at these quickly. Here's an astrocyte. These are by far the most abundant neuroglia in your central nervous system. And the astrocytes are important in controlling the chemical environment in which the neurons are functioning. They are an important part of the blood-brain barrier. In fact, the astrocytes, in this diagram, it's showing the astrocytes sending its projections to cover the capillaries that are supplying blood to the nervous tissue, and they can control the permeability of those capillaries very tightly, and therefore controlling what compounds get through to the nervous tissue. So they serve a very important regulating function, nutritive function for the neurons. Microglial cells are very small. They have kind of an elliptical nucleus and have a lot of these branch projections. The microglial cells are basically the only cells in the nervous system that are in charge of defense. So they're akin to your immune system cells, but they're not derived from immune system cells per se. Ependymal cells are going to line the central cavities of the ventricular system that we'll talk about in later lectures. And the ependymal cells are important for the production of cerebrospinal fluid, or CSF, a very important part of the nervous system. Here's a quick look at this ventricular system that I'm talking about. You have a central canal in the spinal cord and a series of ventricles that are spread throughout the inside of the brain. The oligodendrocytes are cells that are going to be responsible for providing myelination of the axons of nerve fibers. And here's a picture of one, and it's showing a single oligodendrocyte sending projections to many nerve fibers and wrapping around many times around these nerve fibers. And the membranes of these oligodendrocytes are rich in special types of lipids called myelin, and that's why the sheaths around the nerve fibers are called myelin sheaths. And the myelin sheaths provide insulation of those nerve fibers, similar to the plastic wrappings of electrical cords, if you will. And this also helps the speed of nerve conduction to go faster. The areas that are heavily myelinated, where you have a lot of nerve fibers that have heavy investment of this myelin sheaths, give the nervous tissue a whitish appearance, and that's what we call white matter. When we look in cross-section of a brain, we see this whitish areas. That's white matter. Those are myelinated axons, and so you can guarantee that you'll see lots of oligodendrocytes in white matter. In the peripheral nervous system, the situation's a little more simple. You have satellite cells which control the chemical environment around the nerve of the neuron cell body, and so they're providing nutrition and controlling the environment that cell is experiencing. And then the Schwann cells are providing the same function in the peripheral nervous system than the function of oligodendrocytes in the central nervous system, which we just saw. The Schwann cells, each Schwann cell is responsible for going around a particular region of a nerve fiber. So compare that to an oligodendrocyte, in which a single cell can surround many nerve fibers. Here, a given Schwann cell, it only wraps around a single nerve fiber and only a certain region of them. So you need many Schwann cells to effectively cover the length of a neuron, as we have here. Here's a closer look at the process of how a Schwann cell provides these multiple concentric layers of myelination. This is what I was talking about earlier, comparing white matter versus gray matter. Here's in the picture on the right, in regions labeled as B, you see that whitish material. That's white matter. And these are dense collections of myelinated fibers. The other thing that you don't have is in white matter is you don't see cell bodies of neurons. You see their axons, their projections, but you don't see the nuclei of neurons. The cell bodies and nuclei of neurons are found in areas of gray matter, which are shown by the letter A on the right, on the slice of the brain. And notice that all the edge, the periphery of this brain tissue is gray matter, as well as some isolated pockets inside, embedded in the white matter. In the case of the brain, the areas of gray matter that are found on the edge or the cortex is the cortical gray matter, because it's in the cortex, the very outside shell of the brain tissue. And the isolated patches of gray matter embedded in the white matter are called nuclei. Not to get confused with the nuclei of the cell. These are just called nuclei, and there's several of them in the central nervous system. In the spinal cord, it's the similar situation. Here you have on the outside areas of white matter, and then the inside, this interestingly shaped, looks like a butterfly-shaped mass of gray matter. The other thing I wanted to point out with this slide is this spinal ganglion. So in the pinkish, purplish stained cross-section of the spinal cord on the lower left, I don't know if you see on the lower left a little nodule, if you will, called the spinal ganglion. And that houses cell bodies of neurons, but now notice that we're away from the spinal cord. We're not in the brain. This is part of the peripheral nervous system. You do find cell bodies of neurons in those ganglia. So cell bodies of neurons in the peripheral nervous system are pretty much restricted to ganglia, whereas the cell bodies of neurons in the central nervous system are found in regions of gray matter, either, as I said, the cortex or these nuclei that we talked about. So let's look at a neuron next. A neuron has a receptive region at the top. It can be very branched, called the dendrites. All these projections are called dendrites. And these dendrites lead to the body of the neuron, called the soma, S-O-M-A, the soma of the neuron, that contains a single nucleus. And surrounding the nucleus, we find dense aggregations of what are called Nissl bodies, N-I-S-S-L bodies. And these are nothing more than rough ER, rough endoplasmic reticulum, that stains really heavily with this Nissl stain that somebody developed. But anyway, these Nissl bodies are very busy producing vesicles full of neurotransmitters, right? So you know neurons are releasing neurotransmitters at the axonal terminals. All those neurotransmitters are made in the soma of the body in the classical vesicular transport fashion that we talked about when we looked at the cell. Here you have in the body of the neuron, you'll find a region that leads to the axon, and that enlarged area is called the axon hillock. And the axon hillock leads to the long axon, and that carries the impulses down the axon. And then at the very end, you have the axonal terminals. They can be branched. And this is the secretory region of the neuron, and it's the part that's releasing neurotransmitters at the synapses with either another neuron or between a neuron and an effector organ, such as a muscle or a gland. Neurons are categorized by structure in this way. You have what are called multipolar neurons. These are probably the most classical shaped neuron, and where they have many dendrites leading to a single axon. In the middle column, we see the bipolar neuron, and these are not very common. These are pretty much restricted to some of the sensory regions in your retina. But you see that there's a single dendrite. It can be branched, but a single dendrite leading to the cell body that then leads to an axon. The other type of neuron is called unipolar, also known as pseudo-unipolar, and it's shown here on the right column. and you basically have a single axon, and the cell body is off to a side, somewhere along the middle of that. And generally we say that the receptive endings are dendrites, but they quickly lead to an axon, and with its axonal terminals. Next, we're going to talk about how a neuron generates and propagates action potentials or nerve signals. In the dendrites and in the body of the neuron, the cells can propagate what are called graded potentials. So these are potentials that degrade over distance. These regions of the neuron have chemically gated ion channels that cause that depolarization to move down the dendrite and body of the axon. But as soon as those signals reach the axon hillock, you find that it triggers voltage-gated ion channels. And those voltage-gated ion channels are also found throughout the axon, the length of the axon. And that is why these regions generate and propagate now action potentials as opposed to graded potentials. So, and the other important point is that the action potentials travel only in one direction. Once they're generated in the axon hillock, they go down to the distal tip of the axons. Here we see a diagram of comparing the chemically gated or ligand gated ion channels with those of the voltage gated channels. So notice that in the chemically gated channels, remember, they either open or close depending on whether they're bound or not to some chemical, in this case a neurotransmitter. And again, we're going to say that we find these in the postsynaptic membrane and in the synapses. So in the regions of the dendrites and the body of the neuron, this is where you have these. These are the channels that are responsible for the graded action potentials. Once you get into the axon hillock and axon, we start seeing more and more of these voltage-gated channels that open or close in response to changes in voltage across the membrane. To discuss how a membrane has a voltage potential across it, we have to discuss what is called the resting membrane potential. But basically, when you measure with a voltmeter the voltage across a membrane, we find that the inside of the cells tend to be more negative relative to the outside. So there's a potential voltage across this membrane. And when we say that we depolarize the membrane, it means that we are making the inside more positive relative to the outside. We're changing the polarity, if you will, across the membrane. In this slide, this is what this is shown. You have a resting membrane potential around negative 70. When you cause something, a signal that causes a depolarization on the membrane, basically what you get is a upward movement of that trace more towards the zero line. Zero line, it would mean that there's no difference in voltage across the membrane. So here you're depolarizing the membrane ever so slightly. If you depolarize it strongly enough, you can make actually the inside be net positive, and therefore this trace would shoot up past zero into the positive side of the curve. We can also make the inside of the membrane even more negative, and that's called a hyperpolarization. So here again you see the resting membrane potential around negative 70. If you hyperpolarize the membrane, you make it even more strongly more negative, and showing us the downward trace here in this graph. That is called hyperpolarization. There are two types of signals that neurons propagate, and these are changes in the membrane potential that will act as signals. Any change can be a signal. But there's two flavors. Here's as I mentioned, the graded potentials. These travel a short distance. They're the incoming signals to a neuron are through a graded potential. And then the action potentials are all or none fast propagation of signals that are long distance traveling down these very long axons. And so here's an example, a diagram of a localized region of depolarization caused by some stimulus, right? Imagine this could be a ligand-gated channel opening, letting sodium in, causing the inside more positive, causing that region of depolarization, which will then spread. And here in the next slide, it shows this happening. So as you get the inward flux of sodium ions will tend to spread out along the membrane, causing the spread of the depolarization. And because the farther you get away from the stimulus, the more leaking happens of ions. Eventually what happens is that signal decays over distance. So the initial depolarization is highest close to the region of the stimulus where it happened. And as I mentioned, this happens in regions of the dendrites or the cell body. But once that signal reaches the axon hillock, it can stimulate the opening of voltage-gated sodium channels, which will then trigger an action potential. For that to happen, that initial graded depolarization has to be at what we call threshold. That depolarization, as it fades, as it degrades with distance, it has to at least be of a certain threshold before it can trigger an action potential at the axon hillock. If it is below threshold, it won't trigger an action potential. If it is at or above threshold, it will trigger an action potential. Once the action potential happens, there's no stopping it. Here we see what an action potential looks like. Here you start at resting state, -70, and the initial place where you have this upward trend up to -55, see that? Right there, this is an initial graded depolarization, graded potential, and in this case, it reaches threshold. And once it reaches threshold, it activates in the axon hillock the opening of the voltage-gated sodium channel. So imagine this is a tracing measured at the axon hillock. So once it happens, there's an increase, a dramatic increase in the influx of sodium due to those voltage-gated sodium channels, and that sends the depolarization way up here into. We did not talk about how this then goes back down, but just know that it does. This part of the curve is called the repolarization, and it continues on a little past threshold. It continues past threshold and past resting membrane potential to achieve what is called the hyperpolarization phase, and eventually it goes back to resting membrane. And this is all happening very quickly. So here this is measured in milliseconds. So again, this is the overall look of an action potential curve. And in the next slides, it shows the sequential propagation of an action potential as measured in this, where this recording electrode is shown in the figure. Because action potentials are all or none, they either happen or don't happen, it's interesting how the body can code for the strength of a stimulus and also how it codes for the strength of a motor output. So it's an all or none response, a 1 or a 0. There's no such thing as a weak action potential and a strong action potentials. You can think of all action potentials being pretty much the same. This sharp upward peak of depolarization that happens, followed by repolarization, a hyperpolarization, and go back to resting membrane potential. The ways neurons can code for stimulus strength is by modulating the frequency of action potentials, so that if a given stimulus is subthreshold, it produces no frequency, produces no action potentials. But if the stimulus strength reaches threshold, it'll produce here in the middle action potentials at a given rate. And if you have a stronger and stronger stimulus, those action potentials will happen more and more frequently, so closer together in time. And we're not going to discuss how this happens either. Just know that this is the main way in which neurons can code for stimulus strength. So here's an overview of the different ways in which nerve signals are conducted. Here again, he shows you in a dendrite, in a typical dendrite, you're getting a graded potential in which the depolarization is greatest at the site of stimulus and it fades over distance. Here in this middle panel, you see an axon, an unmyelinated axon, in which the action potentials are jumping, well, they're moving from voltage-gated ion channel to voltage-gated ion channel in a sequential fashion. And notice that there is no fading of the signal, because as soon as it reaches those voltage-gated channels are so close together that that signal propagates very quickly. What happens with myelination in the bottom panel here is that in the regions between the Schwann cells, those are the naked regions where there's axon membrane exposed to the outside, we found dense concentrations of voltage-gated channels. These little nodes are called the nodes of Ranvier, the regions between Schwann cells. And what happens is that the voltage, the current, jumps from node to node in a very quick fashion. And this has the effect of greatly speeding up the conduction speed of action potentials down the axon. We can classify nerve fibers based on their diameter, the degree of myelination, and therefore the speed of conduction. So we didn't go into too much detail about this, but the larger the diameter of an axon or of a nerve fiber, the larger the diameter, the faster that it can conduct an action potential down that axon. We already saw how myelination affects speed of conduction. The more myelinated a nerve fiber is, the faster it can conduct an action potential. And so we see that in your body, you can classify most nerve fibers or nerves into these three different groups. Group A fibers are the fastest of them all. They're large diameter and myelinated and can conduct at speeds over 300 miles per hour. Grade B are the in-between ones. They're lightly myelinated. They're of an intermediate diameter, and they can carry signals around 30 miles per hour, so 10 times less so than group A fibers. Last but not least are the group C fibers. These are the smallest diameter fibers. They're unmyelinated, and that means that they conduct the action potentials at the slowest speed, which is around 2 miles per hour. So still fast. It's fast enough for their need. They're typically found in a lot of autonomic control of autonomic functions. And so there you have it. Those are the three types of nerve fibers based on diameter and degree of myelination. We didn't go into too much detail about the synapses that neurons make. We briefly looked at them when we talked about the neuromuscular junction. But you know that synapses are the regions where neurotransmitters are released and they affect the postsynaptic membrane in whichever way they're exciting it or inhibiting it. And I wanted to point out that here that you can have synapses between neurons happening in different regions. You can have the synapses happening within the body of the neuron, in which case they're called axosomatic synapses. And if their axons are connecting to dendrites, we call it axodendritic synapses. And in very few rare cases, you have synapses that are very close to the axon itself. So you have almost an axon synapsing on an axon, generally close to the axon halic region, and those are called axoaxonic synapses. By far, axosomatic and axodendritic synapses are the most abundant. Here is a close-up diagram of a synapse. You should know that when the axon potential reaches the axon terminal, that action potential stimulates voltage-gated calcium channels as opposed to voltage-gated sodium channels. So these voltage-gated calcium channels let calcium in, and calcium then through mechanisms that we did not talk about cause the fusion of neurotransmitter-laden vesicles with the plasma membrane of the axon, and when that happens, those vesicles empty their content, empty the neurotransmitter in the synaptic cleft at the synapse. Once the neurotransmitter is released into the synaptic cleft, it can bind to ligand-gated ion channels and depolarize the neuron down the line. And I want to note that because the rate of diffusion is relatively slow process compared to how fast action potentials move down an axon, we say that this is the limiting factor in the speed of conduction down a chain of neurons. And this concept is called synaptic delay. So synaptic delay is a very important product of the fact that you have these synapses that depend on diffusion of neurotransmitter across the synaptic cleft for the signal to be carried on down the line.